Your cell culture is contaminated. But what exactly contaminated it, and how do you get rid of it? Bacteria can make your medium cloudy. Fungi may appear as floating particles or filaments. Yeast can multiply rapidly and change the appearance of your culture. For these visible contaminants, the safest approach is usually to isolate and discard the affected culture. Then clean and disinfect the workspace and equipment using your lab's validated procedure. But the most dangerous contaminant may be invisible, Mycoplasma. It may not make your medium cloudy, but it can alter cell growth and experimental results. If Mycoplasma is confirmed, don't simply add antibiotics and continue working. Isolate or discard affected cultures. Test other cultures and relevant materials. Identify the source, and perform the appropriate validated decontamination procedure. Then test again before restarting routine work. Because contamination isn't solved when the cells look clean. It's solved when you know the source is gone.